Hello students, I am Dr.R.P.Oviya from the Department of Biotechnology, School of Bio and Chemical Engineering, Satyabama Institute of Science and Technology, Deemed University, and here we are looking into what are all the tools that are used in genetic engineering. In the previous video, we saw about how SDS page can be performed, and here we will see how Western blot can be performed. Basically, ELISA and Western blot works on similar principle, whereas in case of Western blot, there is SDS page electrophoresis should be run and the gel is now used to transfer into a membrane. Whereas in ELISA, the antigen is directly embedded into the 96 well plate or the antibody is directly embedded into the 96 well plate, which is a direct method, whereas in Western blot, SDS page electrophoresis must be performed. So, what is Western blotting? Western blotting is transfer of proteins into a membrane. The membrane can be a polyvinyl PVDF membrane or a nitrocellulose membrane. Here we can see an example membrane where the blotting system can be set up. From the gel, the proteins are transferred into a nitrocellulose membrane. Number one is isolation of protein. We have to get the protein sample out of, for say, here I have given an example as fish muscle, where we can also say a plant protein or a mammalian cell culture protein. So, in all these ways, there are both physical and chemical method that can be used in lysing the sample. Once we get the lysed samples, we can even isolate a purified form of protein or a lysate which contains a whole total protein content can also be run on an SDS page. So, we saw in the previous video how SDS page can be done. The amount of protein that can be loaded in a well can be around 40 to 60 μg per well. So, when we add more amount of protein, then we can even obtain more amount of proteins that can be transferred into the nitrocellulose membrane. So, once we have performed the SDS page, do not do the staining using Kumasi and directly go for the transfer. To set up the transfer, there is a sandwich method. The sandwich method ensures that along the cathode side we have Wattman filter paper kept and above that there is gel and above that membrane is present, such that from the negative the protein gets transferred to the positive side, so that the contents of the proteins get embedded to the membrane side. Post the transfer, it is called immunoblotting, where we can add a particular antibody and do immunoblotting, where we can check for specific protein expression in the membrane. So, this is a stained gel where you get all the bands present in it, whereas here it is an unstained and only the specific antibody can be seen using this method. Whereas to ensure whether the membrane is got transferred, whether there is protein content in it or not, we can do a simple method called Ponceau staining. In the Ponceau staining is like a rubber. You can check for the stain, look into the bands, and once you confirm there is protein transferred into the membrane, you can wash it with water and ensure there is no stain present in it, and then go for antibody incubation. So, post antibody incubation, we can check for specific target protein. This the principle works for similar for both ELISA and Western blot. Here we can see an example SDS page how polyacrylamide gel electrophoresis is done, and once it is done without staining it has to go to Western blot, whereas to check only when there is presence of transfer we can do Ponceau staining on the membrane and then wash it well, then go for antibody incubation. So, once that the gel is trimmed, we have to trim the excess gel, the excess wells and the excess basement can be trimmed, and only the target gel can be used for sandwich setup. In the sandwich setup, in a semi-dry blot, there are two types of blot where we can use semi-dry or wet transfer. In the wet transfer, the transfer happens in the running gel buffer, it is completely filled with the buffer. Whereas in semi-dry, only the buffer is being sprinkled on it and the time taken is very much lesser when compared to wet transfer. So, here the cassette is a semi-dry type. In semi-dry type, we can directly place the filter paper and above it we can keep the gel, and then the membrane, above it we can keep the filter paper, ensure there is no air bubbles. The air bubbles can interfere with the transfer of the protein, and that is why we have to ensure the sandwich is made without any air bubbles. Once we place everything on it, we have to press the sandwich firmly such that we can remove the excess water and excess air bubbles on it. Then we have to lock the cassette and then switch on the electricity. The volts can be kept off very low. The voltage depends on the time. Like suppose we are going to transfer a higher molecular weight protein, then we need more time to get transferred to a membrane. Hence we get lower vols. So for high molecular weight protein, we have to keep lower vols and longer time at 4 degrees Celsius. Example, a 200 kilodalton protein, we can keep at 50 volts for 4 hours at 4 degrees Celsius. Whereas when it comes to a smaller molecular weight and a quick transfer, even the recently developed methods, we have a quick transfer where we can even do at 100 volts for 5 to 6 minutes. So once we have done, we will get the membrane out. Now we have checked for the Ponchus stain, and then we can go for blocking. What is blocking? A blocking refers to an agent that agent can block the binding of any antibody non-specifically to the target. We have to ensure that the antibody that we add is specifically bound to the target, and hence blocking will ensure that no non-specific binding happens. Here the blocking can be used using bovine serum albumin or skim milk powder. Generally skim milk powder is preferred to ensure that there is no non-specific binding present. So 5% skim milk powder can be prepared in phosphate buffer saline, and this also contains a Tween 20. It is a non-ionic detergent, and this detergent will not be giving harm to the protein, and it's a mild detergent, and hence this can be used in disrupting non-specific antigen-antibody interaction and use it for specific target identifying. So once we get the antibody, we can incubate the antibody with the blocking. The antibody is added along with the blocking solution and ensure it is incubated for 1 hour or even overnight. That depends upon the type of antibody we are using. So to get a quick binding, we can use even 1 hour at room temperature or an overnight incubation at 4 degrees Celsius. So the difference between ELISA and Western blot will be both have same principle, same method where there is antigen antibody interaction, whereas in ELISA we use a 96 well plate format where the antigen is bound to the polystyrene coated plate, whereas in Western blot the gel is run and it is transferred to the membrane, and in the membrane the protein is embedded. Now to the both that method, first step is blocking. The blocking solution can be 5% PBS solution containing bovine serum albumin or skim milk powder, and then do wash thrice and then incubate it with primary antibody. Then post that we can incubate it with secondary antibody. The secondary antibody can be conjugated with HRP, or it can even have any other conjugation, example ALP or colorimetric method. So once secondary antibody conjugation is done, we can visualize the band. Whereas here we have a substrate added and we can see difference in the color formation. Whereas in Western blot, if it is an HRP conjugated one, we can use chemiluminescence, and an apparatus called gel dock can be used to visualize the target specific method. So this is how a final answer will look like and how our target protein is present in each of these samples. So this is an example of blot from unstained gel, and this is a stained gel. Whereas in ELISA, it's a direct method, and I told you about the primary antibody incubation and secondary antibody incubation. Whereas in ELISA, the washing plays a major role, where the washing must be done such that there is no excess particles present or there is no non-specific addition. That's why there is washing for thrice and every 5 minutes. So this is a simple ELISA method where we have a simple spot test where we can check for presence of HCG antibody, and we can look into the pregnancy test whether if it gives a single band it's a negative, whereas only in control it shows a positive, whereas when we get a 2 bands it's called positive, while here we can say that is pregnancy positive. It's a simple kit based method which works based on the principle of ELISA. So the main difference between an ELISA and a Western blot is both are qualitative, whereas ELISA is more quantitative while Western blot is semi-quantitative. Though it is semi-quantitative, it's a gold standard method where any scientific community will agree with the results that we get for Western blotting when compared to ELISA. ELISA is not lesser time consuming when compared to Western blot, whereas Western blot is performed post SDS page transfer, and hence it will take even 8 hours to 10 hours. We don't need to run electrophoresis, where here it is more physically tedious, where we have to run electrophoresis, extract the protein and then do. Here it is cost effective. The cost to run an ELISA is lesser when compared to Western blot, where we need costlier membrane as well as a Wattman filter paper and as well as all the reagents that are required. So here it is a costlier, but it is a good gold standard method compared to ELISA. I hope you understood both the methods, and now you can think about which method is suitable for what kind of research purpose. Thank you.